Good morning or good afternoon. Welcome to the Integrative Genomics session and welcome to Bioconductor. We will start the sessions shortly, but just a few housekeeping items. If you have questions for the speaker, please enter them in the Q&A tab. You'll have the ability to upvote questions for the speaker to answer. If you'd like to answer your questions live, please do just raise your hand. We use the raise the hand feature and we will bring you on stage. And there's a variety of emoticons, the little things that make the pretty pictures, available if you want. If for some reason you have to leave early, we will have a video of this session and all the sessions. Now, let me introduce you to our speakers. First of all, we have Ulisse Chiodi. She's a postdoc at the Institute of the Biomedical Technologies Institute of Biomedical Technologies at the National Research Council in Milan in Italy. And she will talk about her package, Ulisse, and our package to go beyond the boundaries of knowledge of molecular pathways. Sounds really interesting. Thank you, Ulisse.
Okay, can you see the screen? Oh, all right. Hello, everyone. I'm Ulisse, and I would like to talk to you about our R package, Ulisse. Pathway analysis is a useful tool to interpret a long list of differentially expressed genes. It's a very widely used tool, but the model used defines a statistic per pathway. In reality, in the cells, the genes inside the pathways interact. They interact inside the pathways, but also between the pathways. And it's important to study this interaction as these characterize the functioning of the cells. There are a few tools available online to study this interaction, so we decided to develop Ulisse, which is able to study the interaction inside the pathways, between the pathways, but also is able to use single-cell data to study the interaction between different cells. Ulisse needs two input, a gene list that may be or not ranked, and a biological network. It has two workflows, one to analyze bulk data, and the other to analyze single-cell data. On bulk data, Ulisse studies the crosstalk between pathways. He studied the presence of a sub-pathway connected component and the crosstalk between different gene communities, and I will explain this better in a few slides. On single-cell data, Ulisse studied the communication between cells cluster and analyzed the cluster of cells by the point of view of pathway crosstalk. All of these analyses calculate a statistic, an empirical p-value associated to this statistic, and are parallelized to reduce the computational time needed for the calculation. But how we calculate this statistic and the p-value? Let's consider this example. Here we have two pathways, B and C, where we want to calculate the crosstalk. In this case, we calculated the crosstalk as to be proportional to the number of links between exclusive genes of the two pathways, and to be proportional to the weight of these genes. The number of links, here we exclude the two shared genes, so in this case we consider only the link between the gene 2 and 6, and the rank we consider, it may be the significativity of the genes. We evaluate empirical p-value by considering the adjacency matrix of the gene network by implementing a permutation approach. We applied the same methods to all Ulisse analyses. I will show you two examples, one for each workflow. So let's consider first the bulk data. here we downloaded mutational data from breast cancer from TCGA. We calculated the number of variants for each gene, and we provided the top 200 gene as a ranked gene list together with string network to Ulisse. Ulisse built the gene network of these 200 genes, and then we have all these functions to analyze the data. First, we have this function to compare different algorithms to find the communities, to choose the best one, because it's important to identify the gene communities as they are gene related and associated to a similar biological function. Then we downloaded data from pathway data from MSigDB. In this case, we used KEGG. And then we have the three functions to perform the interaction analysis of Ulisse. The first analysis is the calculation of crosstalk. Here the genes are grouped into pathways, and by using the gene network, we calculated the crosstalk between pathways, and we obtain a table like the one you can see, where we have the pathway, the score of the interaction, some descriptive value, and the p-value. By considering only the significative interaction, we can build a pathway crosstalk network where we have the pathway connected by the crosstalk, and we can represent it also like an heat map. The second analysis is the study of the interaction inside the pathways. So Ulisse built a network for each pathway and identify the sub-pathway as a connected component. I show you here only one example of a calcium signaling pathway. This function returns to output. One is a list with the association of each gene to a connected component, and another table is a table with all the pathways. Here I show you only this calcium signaling pathway with the score and the p-value associated to the subcomponent score. The third analysis on bulk data is the crosstalk between pathways of different community. In this case, the genes of the same communities are grouped into pathways, and the crosstalk is calculated only between pathways of different community to study the interaction between different biological communities. The output is that we obtain is a pathway gene association list for each community to have a detailed description of which gene participate to which pathway in each community, and a table like the one we obtained for pathway crosstalk, we have seen for pathway crosstalk, but with the indication of which community we are considering the interaction. Again, the score and the p-value, and by considering only the significative one, we can obtain a pathway network, and we can represent it like an heat map. On single cell data, we use single cell RNA-seq from breast healthy tissue from Nguyen et al. We use CELLULAR package to analyze the data and obtain cluster of cells and differentially expressed genes for each cluster. We provided these genes as a ranked gene list to Ulisse together with the string network. In this case, the rank we used is the -log10 of the FDR, and then we used Ulisse function to download the ligand receptor interaction data from Omnipath, the KEGG database again here from my SigDB, to study then the cluster with by the point of view of pathway crosstalk. I will not show you in detail the results of this function, as we obtain a table like the one obtained for pathway crosstalk on bulk data, but with the indication of the cluster where we are studying the interaction, and we study the communication between cells. In this case, Ulisse used the differentially expressed genes together with the network and the data from ligand-receptor interaction to build a network where we have only genes interacting, that only ligand-receptor genes interacting and allowing only ligand-receptor or vice versa interaction. We have two output, one with the detailed description of the communication between the cluster with all the genes involved and their role, and another with the cluster communicating with their score of the communication, the p-value associated with the communication, and by considering only the significative one, we can build a cell network where we observe clusters connected by a signaling that is significative. What we can say in conclusion, with Ulisse we provide a unique place where to study interaction between genes inside the pathway, but also outside the pathways, between pathways. Moreover, we are able, with Ulisse, we are able to apply this analysis to single cell data together with the study of pathway crosstalk, but with also the study of the communication between clusters of cells. What we plan to do in the future is to integrate in Ulisse analysis drug-gene interaction information to provide a tool to study if crosstalk may influence drug treatments. We are currently testing all the method I've shown you today, and we are writing the vignette, and our package will be available soon on GitHub and Bioconductor. Sorry. Thank you for the attention.
Thank you. Do people have questions? We can ask them to Alice. Please post them in the Q&A. Can I ask one question there? Where you're looking at the ligand-receptor interactions for the single cell data, this is like really important to try to work out which cells are talking to each other. Were you able to compare your findings to maybe using cell phone DB or some of the other tools that people are using in single cell?
We are not tried it like now. We are testing the method, but I'm sure we will compare it to other available tool. I've read all the literature that are available for cell signaling. In Omnipath database for ligand-receptor interaction are present also the data from like cell phone DB or other database. So we decided to develop the tool as to be able to download the data from online resource, just as to be, I mean, to be able to allow the user to use data that are currently available on widely resources.
And a second question there is, when you're looking at the cell-cell interactions, particularly with the single cell data, a lot of the times individual cells will drop the gene expression. So are you how are you sharing the information around the cluster, and how's the network being used to facilitate that?
We there's a function of Ulisse are built to work on a cluster of data, so not on on the single cell of the sequencing. We obtained the differentially expressed genes using Sera. In this case, especially in this case, I used the genes of single comparison between clusters. So, I mean, like the differentially expressed genes between cluster 1 and 2, only the genes that are regulated, more present in cluster 1 and the one more present in cluster 2. So it's very specific, but I think that the user will think about what will be the best way to obtain the differentially expressed genes for each cluster. If there may be more important to obtain the one that are specific or of a cluster or specific of that interaction, that communication between the two clusters.
Cool. Okay, I think we should. So this isn't. I quickly checked. I couldn't see this up in GitHub at the moment. So
Yeah.
it's not. Is there a preprint available, or if people are interested in this, should they just contact you by email? What's the best thing?
We didn't allow. I mean, it's not already available because Ulisse is not fully developed. We need to find to check to try all the methods to then be able to publish it. We are testing all the methods. We are writing all the vignettes, all the tutorial, and we plan to publish it by the end of the year, I hope, or at least submit it by the end of the year. But if any user wants to be up to date to Ulisse, they can contact us or contact me by email, so we may be able to have a feedback, give them a feedback, and inform them if they want to. Inform them when the package will be available.
Well, I look forward to it being on GitHub soon.
Yeah.
Okay, let's move to the next speaker. The next speaker is Ian Smith. He's a graduate student in Benhaven Consulab at the University Health Network at the University of Toronto. And he's going to talk about RADIOGX, a package for integrative analysis of cellular features and radiosensitivity in cancer. Thank you, Ian.
Student in the Hybe Keynes lab at the University of Toronto. And today I'll be sharing with you RADIOGX, a package for the integrative analysis of radiosensitivity data. The underlying motivation is understanding the efficacy of radiation therapy. Radiation therapy is standard of care in many cancer contexts, but it's highly variable. It's difficult to determine whether or not a particular patient will respond to radiation therapy. So the goal is to identify biological features that are predictive of radiosensitivity and whether or not a particular tumor or particular cancer can be killed by ionizing radiation. And we use preclinical models like cancer cell lines, and ultimately the goal is to inform patient care. This falls within the field of radiogenomics. Unfortunately, radiogenomics has become a bit overused, and now it has a definition relating to medical imaging, but the original definition is the relationship between biological features and radiosensitivity. To understand radiosensitivity, there are a variety of large-scale studies. For example, Yard et al. 2016 published this paper, A Genetic Basis for the Variation in the Vulnerability of Cancer to DNA Damage. Their method was to take a panel of model systems, in this case cancer cell lines from the Cancer Cell Line Encyclopedia, treat them with a variety of radiation protocols and measure the viability, whether or not the cell line was killed by the radiation. In this case, the y-axis here is the integral of the survival as a function of dose, so whether or not the cell line is resistant or sensitive to the radiation, and the x-axis is a range of different tissue types. So this is a powerful resource for understanding the relationship between the underlying biology and the radiosensitivity phenotype. And certainly papers and studies like this are becoming more common and more available. So it's critical that this data be accessible and more usable. So this leads to the RADIOGX package, which is a computational toolbox for radiosensitivity analysis. RADIOGX, which is available on Bioconductor, combines radiosensitivity data in the form of radiation dose response curves and a variety of molecular features depending on the study, like gene expression data, mutation, copy number and epigenetic data. It also brings analysis tools for providing a unified analysis of both of these data types, which is critical when you're trying to analyze data across a range of different studies. The RADIOGX toolbox was published in Cancer Research. The link is below. The sort of basic unit of RADIOGX is an S4 class called a radio set or an R set that serves as a container for unifying this radiosensitivity data and molecular features. And finally, RADIOGX integrates very well with other elements of the BHK Lab software ecosystem for analyzing model systems. For example, PharmacoGX for analyzing drug response data, and ToxicoGX for analyzing toxicity. So the radiation data, the radiosensitivity data in RADIOGX takes the form of dose response curves. So on the left we have an example figure. The x-axis is the radiation dose in gray, so ionizing radiation, and the y-axis is the log 10 viability. So smaller values on the y-axis indicate that the cell line was killed by that particular dose of radiation. Each one of these lines is a different cancer cell line, and as you can see, there's a range of responses. Some cell lines are relatively flat. Their viability is flat as a function of different doses, indicating resistance to radiation, and other cell lines are sensitive, indicating by a precipitous fall in their viability. RADIOGX also incorporates molecular data, for example, gene expression data. And on the right, we have a t-SNE plot from the Yard et al. Study of gene expression data. Each dot represents a different cell line, and the colors indicate different tissues. Unsurprisingly, there is a tissue-based pattern to gene expression data. So the radiation dose response curve, one of the analysis tools in RADIOGX is summarizing the dose response curve. So the sort of standard is to model dose response curves using what's called the linear quadratic model, where you have this expression. S is the surviving fraction of the cell line, so how much was not killed, and it can be represented as an exponential with a linear term as a function of dose, d, and a quadratic term with dose. And these coefficients alpha and beta characterize the sensitivity of a biological system to radiation. So on the left here we have two different cell lines, SNU245 and SKES1. Each one of these dots is a point measurement of the survival as a function of dose, and these lines represent the linear quadratic fits. Finally, the overall characterization of a cell line, the overall radiosensitivity, can be summarized by taking the area under these fit curves. And large values of the area under the curve indicate resistance to radiation, and small values indicate sensitivity. Just to provide a little demonstration of some of the analysis capabilities of RADIOGX, we used RADIOGX to formulate a method for identifying radiosensitizing agents. So to begin, we took the R set, the radio set from Yard et al., and summarized the gene expression data using reduced representation, pathway level activations to reduce the dimensionality of the space into relevant biological features. Use that to generate a radiosensitivity signature, and then use that signature to identify candidate drugs from the connectivity map. Our setup was fairly simple. We took this reduced representation of the gene expression space. So this represents a big matrix where the rows are pathways and pathway level activation, and the columns are cell lines from the data set. And for this analysis, we used Plier, the pathway level information extractor, which is a different R package. And we constructed a linear model predicting the area under the radiosensitivity curve as a function of these pathway level features. We did sort of standard machine learning practices with cross-fold validation and an elastic net regularized linear model. So the results, the linear model was modestly predictive on unseen data. So the x-axis here indicates the predictions on cross-validated, so held-out data, and the y-axis is the true values of the area under, or the measured values of the area under the dose-response curve. Each one of these dots represents a cell line, and the overall correlation for pan-cancer radiosensitivity was Pearson of 0.44, which is certainly statistically significant. On a biological process as complicated as radiosensitivity, this is fairly informative. Also, the analysis was able to identify pathways and molecular features that in particular were associated with radiosensitivity, radioresistance. There is additional analyses in the RADIOGX publication, but this is just to illustrate that this data integrates very smoothly with other packages and tools. We then used our radiosensitivity signature from the model to query the connectivity map, a resource of differential gene expression signatures from compounds that was published in 2006. And the x-axis here is the enrichment of our signature. Y-axis is negative log 10 adjusted p-value. And each one of these dots represents a different compound from the connectivity map. So some of the highlights, these are candidate radiosensitizers that are putatively associated with radiosensitivity. We're doing follow-up experiments now to determine whether or not any of these actually induce radiosensitivity, which is sort of the goal of the project. So to summarize this analysis, we used RADIOGX to characterize a signature of radiosensitivity in gene expression space. We use that signature to identify candidate radiosensitizing therapeutics. And we integrated the radiosensitivity data from RADIOGX with pathway level tools and the connectivity map project. So to summarize, RADIOGX is a useful toolbox for analyzing radiogenomic data, which is an important frontier in cancer research. It's available on Bioconductor, and it's an open source package. I'd also like to shout out Orchestra, which is a platform from the BHK lab for making public multimodal data generally available. There will be a short talk this afternoon at 1:30 by Anthony Mamaliti and Minoru Nakano in the Infrastructure and Programming Short Talks workshop. And some of the data that are available on Orchestra include radio sets and RADIOGX data. So thank you all for all your attention, and I'd like to thank my collaborators and people from the lab who contributed to this research. This is a recording because I'm not able to be available during this session, but if you'd like to communicate, please reach out to me via email or on Twitter. Thank you very much.
Thanks a million, Ian. If people would have questions for Ian, I'm happy for them to be posted on the Q&A here. And Chris Eccles, who will be speaking next, is actually also from Ben's lab at the University of Toronto, and he may be able to address some of the questions. So please post your questions in the Q&A. I am not going to directly ask questions, Chris, of this, because I know this isn't your project, but it's really interesting to see how the lab have taken some of the fantastic methodology that have been applied for looking at drug response curves and use these to apply to radiology. It's really exciting to see and to look at potentially the overlap in drug sensitivity and radiology sensitivity is really exciting move for oncology, I think.
Absolutely. I'd like to also call back to last year's Bioconductor conference, where we gave a workshop on combining PharmacoGX, looking at drug sensitivity signatures, RADIOGX, looking at radiation sensitivity signatures, and the Ziva package, which is looking at drug sensitivity signatures specifically in PDX models. So I think that if you're interested in RADIOGX and in the talk that was just given, that workshop would be something good for people to check out.
Yeah, I think that's really exciting, and I think the Ben Haben lab and everybody there in Toronto have been really, really good supporting Bioconductor and putting all of your data, resources, and tools into Bioconductor. So thank you so much for that. I'm not seeing questions. People are probably going to email him directly. So let me introduce your talk, which is very exciting. So Chris Eccles is actually a full-time software developer in Ben Hagen's lab at the Princess Margaret Cancer Research Centre. And he's going to present PDATK, an R package for molecular classification and survival prediction in pancreatic ductal adenocarcinoma. Thank you so much, Chris.
Thanks for having me. Is everyone able to see my screen?
I can't see it yet.
Can't see it yet. Okay.
Oh, now I can.
Okay, perfect. So as mentioned, my name is Christopher Eels. I'm a software developer in the Benjamin Habekins lab. We're located at Princess Margaret Cancer Research Center, part of the University Health Network in Toronto, Ontario. Today, I'm going to talk to you about the pancreatic cancer overall survival predictor, or PCOS, which is one of the methods available within the PDATK package. So as an introduction, I linked my slides in the chat. They're also available at this link here. And yes, so if you want to follow along, please do. So what are we discussing today? We're going to talk about the Pancreatic Cancer Adenocarcinoma Toolkit, or PDATK. That's a new bioconductor package published by our lab earlier this year, written by myself. And more specifically, we're going to learn how to train and utilize a pancreatic cancer overall survival predictor model. And the PCOS model makes predictions to stratify patients into good or bad prognosis. For this presentation, good prognosis is defined to mean overall survival greater than 1 year, but this might be different for different cancer types. We hope this tool may be useful for other researchers and clinicians. So as a presentation outline, we're going to be replicating part of a study from our lab, Sentu et al. 2019. The reference is at the end of the presentation. And in doing so, we're going to learn about how to train a PCOS model from existing data, specifically RNA sequencing and or microarray data with known survival outcomes. Second, we're going to use the trained model to make risk predictions for new patient cohorts. We feel like this is an area that's interesting for follow-up research. And finally, I'm going to demonstrate how to calculate and visualize some model performance statistics using independent patient cohorts with known survival outcomes. A more complete analysis is available in a PDATK vignette linked in this slide, although it only uses a subset of the data. So replicating Sentu et al. 2019. So the original publication is called Meta-Analysis of 1200 Transcriptomic Profiles Identifies a Prognostic Model for Pancreatic Ductal Adenocarcinoma. It's available at this DOI. The paper pioneered the PCOS method, and it also demonstrated that it can match or outperform other published signature-based risk prediction models, at least at the time of publication. So the data from this study is available. It's been curated and released as the RADIOGX Pancreas Bioconductor Data Package. It contains 15 published pancreatic cancer patient cohorts. These are either microarray or RNA sequencing based profiles of patients. And 11 of 15 of cohorts have patient survival metadata. So that would be survival time as well as overall survival status. Okay, so to get started using RADIOGX Pancreas and PDATK, I have some code here that will install your dependencies. As you can see, I've also included BioC Parallel. That is because we use that package to control parallelization of code execution, and you'll need that package if you want to modify any of the parallelization parameters. Okay, so the first step in this analysis is to load our data sets. We can do this with the load pancreas datasets function from RADIOGX Pancreas, and this will download several gigabytes of data to your experiment hub cache. The next step is to extract the summarized experiments containing our patient cohort data. As you can see here, I've listed the cohorts which have available survival data. I do a little regex to fix up their names, and then I subset to our cohorts with survival data. The next step is creating a cohort list. This is a class included in PDATK, and a cohort list is just a simple list of survival experiments. and a survival experiment is just a wrapper around a summarized experiment with some mandatory survival metadata columns. So you'll see here, I'm converting our summarized experiments into survival experiments using the survival experiment function, and I specify which columns have our survival time and our overall survival status. So the next step is to split our data into training and validation sets. The first step in this is to find common genes. This can be done with the find common genes function on a cohort list. That will return a list of common genes between all patient cohorts. The next step is to subset those cohorts to common genes. That can be done with the subset function, and that will actually subset all items in the list. Next, we're going to extract our training data from our cohort list. In this case, we're using the ICGC data that we've collected on pancreatic ductal adenocarcinoma. This includes microarray and RNA sequencing data. So once we've extracted our training data, we'll subset that out of our validation data. And then we'll drop censored patients, being patients who dropped out of the study before we could make an observation. So we're not sure if they're alive or if they have passed, so we need to remove them from the data. You can do this with the drop not censored function, which I will note should be renamed to drop censored and will be in an update to the package coming soon. To continue, we'll find common samples in our training data with the find common samples function. Again, we will subset our ICGC training cohorts using the subset method to select the common samples. And then we'll take the anti-match of all of that with the invert equals true argument to have some holdout data from our training data that we can also use for validation. So we'll merge that with the remaining 9 validation cohorts into our validation cohort list. That is a list of survival experiments, one for each patient cohort. And then we will drop ICGC-seq from the validation data because unfortunately it only has 7 patients left after intersecting on samples, and this will break some downstream code. Okay, to the interesting part, setting up a PCOS model. So we've included an S4 class in this package that automates a lot of the analysis for you. So all you need to do is set a random seed and then call the PCOS constructor on your training cohorts. Specify a minimum days survived. This indicates what is called good survival. So anything greater than or equal to 365 days is considered good. And then we capture our random seed to ensure that our model is reproducible. So a PCOS model is a risk prediction method which uses an ensemble of k top scoring gene pair binary classifiers to vote on a patient's prognosis. So this is a supervised learning method that uses survival metadata in the training data to learn important prognostic features based on these survival labels. The PCOS score is defined as the proportion of binary classifiers which vote good for prognosis out of our ensemble. So for training our PCOS model, just as a note, this will take up more than 32 gigabytes of RAM, especially if you have multiple cores. So you may need to modify your parallelization settings to run this, but it's simply calling the train model method on our PCOS model. In this case, we're doing an ensemble of 1,000 binary KTSP classifiers, and we are filtering those 1,000 models to a minimum accuracy of 0.6. This is technically a free model parameter, but we found 0.6 is good in testing with our data as a cutoff. So that means that we only include binary classifiers that have a minimum prediction accuracy of 0.6 on our survival data. So this is the part that may be interesting to clinicians and also could be interesting for follow-up research. We can take our trained model and make predictions for prognostic risk on either a cohort list or a survival experiment. So in this case, the first argument to the predict classes method should be a survival experiment or a cohort list if you have multiple cohorts. The second argument should be your trained PCOS model. And not to worry if you don't have your own training data, our pre-trained model can be downloaded from the link in this slide. So I guess the question that some of the astute audience may have noted is, what if we only have RNA sequencing or microarray data without survival metadata, which would be the case for something like a prospective cohort? No problem. We just have to add some fake metadata, and risk predictions are still available. We plan to correct this in a future update so that a summarized experiment can be used directly to make prognostic predictions on patient cohort. So risk predictions with a PCOS model. So for the first step, we need to load your data as a summarized experiment. Here, I'm simply taking one of our validation cohorts, the UNC cohort. I'm coercing it back to a summarized experiment as an example. The next step is to add some fake metadata, and this will allow you to convert it to a survival metadata. So for this, you need to add the survival time and event occurred columns to the column data. That's the sample metadata of your summarized experiment, and then convert it to a survival experiment. Once that is complete, we are ready to make our risk predictions. So as I mentioned before, your survival experiment will be the first argument to predict classes. Your trained PCOS model will be the second, and then the risk predictions will be stored in the column or sample metadata of the risk predictions that are made. So for our validation calculations, we assume a PCOS score greater than 0.5 predicts a good prognosis. That is, more than half of our models vote good prognosis. And this is required to ensure that we maximize the statistical power of our tests in validation. In clinical applications, a stricter cutoff may provide more certainty of prognosis. And this could be a good area for a follow-up study. So here is an example of our risk predictions. You can see the sample column on the right. The PCOS score ranges from 0, being no models predicted good prognosis, all the way up to 1, being all models predicted good prognosis, as you can see in this table. And that is it for risk prediction. So to recap, for predicting risk for new patient cohorts, you will, 1, load your patient cohort as a summarized experiment. 2, add some fake metadata to allow conversion to a survival experiment. And 3, you will make your risk predictions. So now that you have risk predictions, what to do with the results is the interesting question. So we feel there is the potential for more research. Applications in other cancer types, i.e. converting the pancreatic cancer overall survival predictor to a cancer overall survival predictor. And nothing in the code prevents you from using other cancer types with this classifier. Development of a targeting sequencing panel for the PCOS gene signature. So you're able to extract the gene signature for this trained PCOS model and could develop a targeted sequencing panel that you use for a prospective clinical trial. And clinical validation of the PCOS score predictions in a prospective cohort. So this is outside the scope of our lab's research. We're purely computational, but if a clinician was interested in collaborating with us, we would love that. So with the time remaining, I'll talk about validating a PCOS model and why this is a reasonable model to use in clinical trial testing. So to validate a PCOS model, you use the validate model method. The first argument will be your trained model, and the second is your validation data, which in case is our cohort list of validation cohorts. So this performs external validation using the remaining ICDC microsamples. So that's our holdout data from our training, as well as the 9 remaining independent patient cohorts. And it will assign a data table of performance statistics, which can be accessed via the validation stats PCOS method. This is the table which is used to generate all subsequent plots. So plotting model performance. The first thing we'll have a look at is d-index. D-index is the log2 of the hazard ratio between the good and bad prognostic groups. It is a measure of relative risk. A higher d-index is better, as it represents a larger difference between the good and bad prognostic groups. 0 is the cutoff for better prognosis in the good survival group versus the bad. So here is a forest plot showing our model performance. As you can see, we got statistically significant results in all but the OO and winter data sets based on the p-values here. And when we combine these data sets for a meta-analysis, we get a statistically significant log D index, indicating that our predictions are doing better than random chance. In this case, much better. So the next statistic of interest is the concordance index. The concordance index is a measure of similarity between rankings. In this case, the ranking is based on prognostic risk from the PCOS score, and the ranking is achieved by the PCOS score as compared to the real observed survival data in our validation cohorts. A concordance index greater than 0.5 indicates better than random performance, where a concordance index of 1 indicates identical ranking, i.e. perfect performance. As you can see, similar to the D-index results, both OO and winter are not significant. We haven't looked into why this is, as we are mostly interested in the meta-analysis. But as you can see here, looking at the summaries, the RNA micro, RNA sequencing, and overall methods have statistically significant concordance indexes, indicating that they do a reasonable job ranking patients by their prognostic status. Finally, we'll look at a ROC curve. So a ROC curve shows the performance of a classification model. An ideal model would have its curve as far to the top left as possible, and the y equals x line represents statistical insignificance, that the model did no better than random chance. So as we look at our ROC curves, we're able to see for the majority of patient cohorts, we have done better than random chance. There is still room for improvement here. And that could be an area for follow-up research. And you can tell which cohorts are significant based on the p-values presented in this plot. So that is everything I have for you. Thank you for your attention, and I'm ready to take any questions you might have. So I will just exit full screen. There are the references if you're interested in reading the papers.
Thanks a million, Chris. That was a great talk from Chris Seals. Chris, one quick question about the PCOS model itself, which was originally developed on a set of 89 cases that had microarray and RNA-seq data. So is the model like stagnant on those data, or because you had a training and test loop as well? I don't quite fully. Can you give a little bit more information on that?
For the validation cohorts, you'll see we had a combination of either RNA sequencing data or RNA-based microarray data. And because we have trained the model on a combination of these two data types, it is able to accept either the RNA sequencing or the microarray-based data. So if a clinician was interested in doing some follow-up research using the PCOS score to look at the prognostic risk for patients that they're treating, then they could either do say a whole exome sequencing array, or they could look at something like a targeted sequencing panel for just the genes that are selected as most predictive in the trained model. And if you look in the PDATK vignette, it shows you how to extract the top features from the model. So you can start doing some things like pathway analysis, which is also included in the PDATK package. Unfortunately, I didn't have enough time to touch on that today.
That's wonderful, and all the software is available in Bioconductor. So with the PCOS model, how many genes were in that signature? And I know it seemed to me that you were subsetting to get a common set of genes between the different data sets, but I'm presuming the PCOS model that was developed on the 89 patients has just a certain number of genes in it.
So we actually subset to common genes in all of our models, and this is to ensure that we don't end up with a gene signature from our training data that is not present in the validation data. It turns out the common genes it was a large number of genes. I think it was like 20 out of 30,000 or something like that. I would have to double check. And then the gene signature used in the classification model is extracted from each of the K top scoring pair classifiers. So it depends how large your gene signature is. I think in this case, around 600 of the models were performed better than the minimum accuracy cutoff at good or bad prognosis prediction, and there were about 5 genes per model. So that would be about 3,000 genes in the gene signature. But you are able, using the top features function, to extract whatever percentage of the top scoring genes that you like. And indeed, if you look at the PDATK vignette, that's exactly what I've done. I took a small subset of the top scoring genes and used those to run an analysis small enough to be submitted as a vignette to Bioconductor. And interestingly, like, the results are pretty close to what we get with the full data set, which show that we are finding genes that are actually interesting and predictive of prognostic risk.
Cool. I actually linked to your vignette in the chat. It
Perfect.
seems that people are quiet this morning on the Q&A. I don't know if anybody has any final questions for either Alice or for Chris or for Ian. If not, I'll wait maybe a little bit a second, but if not, I'll start thanking our speakers on this wonderful session. So Alice from the Institute of Biological Technologies in Milan in Italy presented on the Ulisse pathway analysis, which looks really exciting. Ian Smith from the University of Toronto presented on RADIOGX. We're looking at radiosensitivity in oncology data. And Chris Eels, also from the same lab at Princess Margaret and the University of Toronto, presented on the PCOS model for predicting outcome in pancreatic cancer. And thank you for joining me this morning in BioC 2021. And I hope you enjoyed the meeting. And I'm going to do a really, really short plug that we're going to be doing a Meet the Community Advisory Board later today, if you would like to find more about how you can give back to Bioconductor and how you can contribute. Okay, thank you. Bye-bye.